Yaw Aniweh is a senior researcher at WACCBIP in Ghana. He's heavily involved in the bioimaging community in Western Africa. And it's a pleasure to have him join us here today, even remotely, to have him tell us about his research and how he uses microscopy in his pursuit of answers in malaria research. So Yaw, over to you.
Okay, thank you very much. Can you hear me?
Yes.
Okay, that's very good. As Karen mentioned, we're more into malaria research, and we are heavily imaging dependent. We use a lot of imaging platforms to get to achieve our aim. Okay, so just a quick background. I did a bachelor's in laboratory technology and then PhD in biological sciences, focusing on molecular genetics and cell biology. Um, that was done in Singapore, and currently I am at University of Ghana at a center called the West Africa Center for Cell Biology of Infectious Pathogens, which is a center at the College of Basic and Applied Sciences. So basically, I am in Accra conducting research. So over the years, I've been working on malaria research, and you will all bear me out that there are different parasites that can cause malaria. We have Plasmodium falciparum, Plasmodium ovale, and malaria. So ovale, we have ovale catisi. That's why you see me put it ovale C, and we have ovale walliceri. Yeah. So these have been shown to cause malaria, especially in the sub-Saharan African belt. It is the dominant, these are the dominant ones, though falciparum is known to cause the heaviest of disease. So over the time, I use genetic approaches to understand the parasite. In the group also, because we deal directly with clinics, we ended up developing a lot of molecular diagnostic tools. And my basic interest has always been in trying to understand what are the correlates of protection in helping us design possible vaccines. And therefore, you'll see a lot of molecular immunological tools being used within my research group. We've been studying the molecular basis of drug resistance as well. So within the group, we have different thematic groups that tackle in these areas. We, as I mentioned, we do vaccine target identification and validation. We do a bit of assay development, but generally we look at the host pathogen interaction. One, from the host end, and two, from the parasite end, to see what are the best molecules in there to be used as intervention, either for diagnostic, for vaccine, or for drug target use. So in our lab here, basically in the field for malaria, you have compound microscopes that are used in determining whether a person is malaria positive or negative. When the parasites come to the lab, we have different imaging platforms depending on the type of assays that is being done and what we are aiming to see and what is the level of resolution we want that allows us to choose. So we have the Olympus BX41 is just an upright. Then we have the Carl Zeiss Aziovert A1. It's an inverted microscope, so it allows us to do different things. Then we have this LSM 800 with Eriscan to try to attempt to do some super resolution kind of things. So just to take you through some of the examples of things that we've been doing, when I joined the University of Ghana, I decided to look back and ask simple questions people who have been exposed to malaria, what are the main candidate proteins of the parasite their immune system have been trained to? And we came across this antigen. We named it PFMAP. That is Plasmodium falciparum merozoite associated apical protein. And we think this is a possible vaccine candidate. We purify antibody from patients and use the antibodies to probe the antigen. And if you look at the late stage of the, let me get a pointer. Can you see my pointer?
Yes, we can see it.
Oh, that's good. So these are late stages of the parasite. We call them schizons. And schizons does have a number of merozoites in them. So if you look at schizon, the staining is more like perinuclear, using that P as an example. But then if you now allow the schizons to release the individual merozoites, like in these cases, you see that the protein gets kind of concentrated at one end, and by co-staining with other markers, we could clearly show that these are the apical end of the parasite. What we could also do is that we could generate antibodies to different regions of this very protein and show that if you do a dose response, it blocks the ability of the parasite to enter red blood cells. So with this, we could also now look at different ends of the protein and ask simple question, which area will be associated with possible protection. And here we can show that the C terminus is mostly associated with protection relative to the N terminus and the middle repeat region. The other thing we could show here is that, you see, when the parasites are intact, it shows the perinuclear, but towards them being released, like you can see in this case, the protein become more concentrated. And in this clear demonstration, you see that it's more apically concentrated. So here with imaging, we can see the dual behavior of the protein. When it is in the intact parasite, it is perinuclear. When the parasites are released as individual merozoites, it becomes apically concentrated. And this is something that is not new to the field, but it helps us in also categorizing the antigen and also in we going down further. The other thing we've been doing is studying drug resistance. And most of the drug resistant genes are transporters, and most of them are ABC transporters. So what we did was to look for some of the ABC transporters that are in the genome but have not been characterized. So that led us to what is called the ABC2-like protein in the parasite. What we could do with antibodies generated to this is that when you stain, so if you look from here down, they mature, and this is within a 48-hour period. So this is time point like 4 hours, this is 6 hours, this is 8 hours, this is 10 hours, 12 hours, 14 and 16 hours post-infection. You could see that the protein is more perinuclear, but then it also shows from here that it is exported to the host red blood cell membrane surface. So from here, clearly, we could have an indication that the protein, much as it is associated with a parasite body, is also exported to the host. And we could do other analysis looking at merozoite, and you see that in the merozoite, individual merozoite, it is clearly perinuclear. And even if you want to look at the merozoite as it enters, this is a whole red blood cell. You can see here, this is the gene of interest, which is in green, is whole. The marker of merozoite is MSP. You see that MSP, as the parasite enters the red blood cell, it sheds the MSP off. So you see that how far it has entered, how much we have the MSP. So clearly, this is an indication that this protein, one is 10, it can play a possible role. We don't currently understand what role it will play, but we think seeing this response. So being on the mesozoic surface, we intentionally decided to use the antibodies to see whether it can block. And one thing that is visible is here. One thing is that there is a saturation. Once you reach there, it's almost plateaus. And it doesn't matter the type of parasite you use, that is the behavior you see. So it tells us that we can achieve saturation, and by achieving saturation, it tells you that you can't go beyond there. So these are some of the things we have been doing. But one particular thing from the host, and there are different proteins, some transmembrane proteins, there are some membrane associated, glycosylated and non-glycosylated. So some of the proteins have been known as receptors used by the parasite. Pathogen, this is clear. And then the Duffy, we know these two as key receptors used by the parasite to enter. So what we have been doing in the group recently is, since we are in a malaria endemic, we could go to the population, look in the people, what are the kind of receptors and what is the diversity of expression. We don't have a uniform expression of receptors. Whoever you are, the one next to you have different expression levels altogether. And here you could see one of the receptors, glycophorin A. This is glycophorin A. This is AB and glycophorin C. The antibodies are designed in such a way that you can distinguish between glycophorin A and C, but then for the antibodies to B, it also recognizes A, so we call it AB. And this is complement receptor 1. Each dot that you see here are individuals. So it tells us that in terms of expression, we all have varied expressions of this. And how does that help us? What we know is that the red blood cell has a membrane. The membrane has an architectural proteins we call the spectrin networks. And you can see here, you see these nodes are junctions. And then we have the mesh, the spectrin mesh. So one thing is that when the parasite is to enter, it has to either cause a disruption in the mesh so that it will be able to enter. And clearly here, if you look at the spectral length, which are biological, it doesn't change. But what you can see change is the size of the mesh, which is one of the things. And that is to tell you that if any of the proteins that anchors the junction is removed, the junction becomes weaker. And if the junction becomes weaker, the mesh expands. And that is a way we believe the parasite uses to enter. And one thing we have been able to show is that one of the proteins we've been working on is able to induce that kind of change in the host that makes it favorable for the parasite to enter. And by entering, it can grow in there. So one of the models we think is that this process is a calcium-driven process. And being a calcium-driven process, it allows the parasite to prepare the host prior to its entry. And these are key junctions that we are looking to developing interventions. Currently, PFRH5 is currently being developed as a vaccine, and we believe that it is one of the promising candidates here. So one thing, just to add a bit to the choice of the imaging module we use is the size of the merozoite is just 1 micron. So you can't just use any imaging system. You could see, but you will not be able to get good information out of that. So that is why we use more of the confocal. But if you to look at the full parasite body, That one you could just use the epifluorescent microscope or even compound microscopes to do that easily. So with this, I wouldn't take too much of your time. I will thank my boss, Gordon Awandari. My team, I have a big team. We call the team the Molecular Genetics and Cell Biology Group here. I will thank everyone on this call for your attention. Thank you.
Yeah, thank you so much. That was fantastic. I'm going to open up the invitation for questions from the in-person crowd first.
Yeah.
Any questions for you? Yes, we have a question in the back. I'm going to ask you to speak kind of loudly, and then I'll just repeat it for the online crowd.
So I wanted to find out how much characterization of the PF map have you done? Yeah.
Cool. Yeah, so that was a question regarding how much characterization of the PF map antibody you've done.
So with PF map, I started with polyclonal antibodies. Now I've generated 20 monoclonal antibodies that currently I'm characterizing. I've expressed the antigen fully, the full length. I've expressed different domains. So the characterization as it stands now, I've seen that it interacts with a receptor on the red blood cell surface. That is what I'm currently fishing out. And I think we've gone far with the characterization of the PF map and looking at this antigen in a cohort, a BEV cohort. We think that individuals that have higher antibodies to this antigen have the less chance of going to the hospital with malaria. So that is where the correlation of protection is coming from.
Cool, thank you. Anyone else? Nothing online, no. Yeah, I have a question sort of following on from that. In terms of the sort of prevalence and variation in this PF map and what you're seeing in terms of other anti-malaria antibodies, given the parasite's tendency to vary its antigen presentation.
Yeah.
Is that something that you took into account in this
No, so
project?
to select candidates, you look at antigenic variation. This is not part of any of the multi-gene families. Number one, this is a conserved protein, but not pan-plasmodiumly conserved. It is only in Lavranian. The Lavranian parasite, we have Falciparum, Yaw Gaboni. Those are the few. And it is thought that the other parasite will have lost it, but because it is essential to these, that is why possibly they keep it. These are some of the schools of thought here. Being Lavranian conserved, the level of polymorphism is very low. So you compare it to other polymorphic antigens, like in Plasmodium, we have what is called PFAMA1, and then the MSPs. They are highly expressed, but they are highly polymorphic. So that is why we don't use those ones as passing candidates, because you will fail. Yeah.
Great. Thank you. That makes sense. One more invitation to the audience for questions. Otherwise, I have one more for Yaw before we wrap up. Okay, I'm seeing no hands raised. So Yaw, my question is a question that I was going to ask other speakers, and then I got you asking another question. So a specific question for you. What made you first decide to pursue imaging to build it into your research? What was it that made you think, wow, I really need to be able to see this parasite?
Yeah, so like you mentioned, the behavior, this is a parasite, it's very small, it does not have a brain, but it's very smart. So it is believed that the parasite has co-evolved with man all this while. So it has built some kind of smartness in how it deploys its antigens. And in malaria, seeing is believing. Though it is a general principle, but I think being able to see, it helps you also find different ways of understanding the mechanism and how you will deploy, assuming you want to design intervention. The other thing is also, aside even intervention, you need to understand the basic biology of it. The size of the parasite. The parasite is of the invasive form is 1 micron. The human red blood cell is 8 microns. So basically, you're looking at two relatively smaller things, though the red blood cell will be easier to see. But how can you really be able to be firm and also be sure of the biology you see? That is why we need imaging, and not just any imaging at all, a higher resolution imaging. So the other imaging modules, I didn't want to make the talk too long. We've done SIM, structural illumination. We've done expansion microscopy, looking at other things in the group. So I just limited it this way, but we're using all the different imaging modules that will allow us to achieve good resolution to enrich our research here. Yeah.
Great, thank you. I'm going to assume that silence equals agreement that this is a good time to wrap up. Thank you everyone for joining us today, both in person and online. Thank you for sticking around even as we ran just over 10 minutes over time. It's been a pleasure to have everyone with us today. I don't know if my Zeiss co-host wants to say anything quickly, or no, we're just going to wrap up. I'm going to share my screen quickly to issue an invitation to everyone to keep an eye out for upcoming sessions, and sort of information will be sent out ahead of time. And we look forward to having you join us again next time as we focus on each of these topics in more detail. Thanks so much. I'm going to end this now.